Hey guys, so we're looking at section 11.4 to kind of wrap up chapter 11 here today. This is called cell differentiation, which is kind of a long term. It's basically referring to cells over time kind of developing specific jobs or functions. So when we talk about differentiation, we're saying essentially that cells are becoming more different from one another for a specific purpose. All right. So in chapter 11, we've talked a whole lot about how we create new cells and the process that kind of involves. But now we're going to look at, all right, so for those created new cells, how do we actually go about assigning them roles and allowing them to fulfill those responsibilities? Okay. So go ahead and get the title as usual. When we look at our objectives for today, we're going to talk about that differentiation process here first, and then we're going to look at these reservoirs of cells that we all have in our bodies, which we call stem cells. And we'll talk about the importance of stem cells, their use in things like medical research, the different types, and why they're maybe a little bit controversial. Okay. So starting with differentiation here first. So our goal here through cell differentiation, when we talk about this in terms of, the human body or any multicellular eukaryotic organism, is going to be to create specialized or what we call mature cells with specific roles. I like to always refer to this as like developing and finding a job, right? It's like finding your role in a community. And for you guys, you're much like these stem cells that we're going to talk about. So you're unspecialized. You don't necessarily have a job. And if you're working a job like part-time, chances are it's not going to be the same job you want to have for your whole life. For some, it may be, but for the most part, when you leave ninth grade year and go on to 10th, 11th, 12th, you eventually end up kind of finding your pathway, right? That's very similar to what happens with cells in any multicellular organism. They start in one position and then, they're kind of driven down a pathway based on a number of different factors. So this occurs during development in any organism that, again, is multicellular. And we're going to try and create cells that have specific roles, okay, specific jobs. So we're looking at 3 different types of plant cells here, and you can see we've got a number of different structures and shapes for these. So this is one type of tissue here. This is leaf tissue, and all of these little green things are chloroplasts that we find inside of plant cells. Here we have a tissue of a plant called xylem, and xylem's job is to absorb water from the roots of a plant from the ground. And you can see what these look like. These are basically empty spaces. These cells contain a lot of empty spaces that allow for the transport of water. And then here is phloem, which you can see is also slightly different. Not quite as many empty spaces, but you have larger channels, it seems like. And phloem is responsible for moving things like sugars made through photosynthesis. So in a lot of ways, structure and shape of a cell has everything to do with what a cell is actually going to do. And so not only is a cell trying to obtain a role, it also needs to figure out what shape it needs to become. So when we look at differentiation in a multicellular organism, we know that this is going to always start during embryonic development. And embryos are something that we know that multicellular organisms through sexual reproduction will eventually start to form after male and female gametes fuse. An embryo is an early stage of development. It comes before in humans, before the formation of a fetus, before the formation of an infant and a baby. So embryo is very, very early. It's essentially where we have a lot of different cells, and it's hard to distinguish actual structures in that organism. But what we know is that based on where a cell starts in an embryo, that determines where it's going to kind of go. All right, so its location in embryo determines how it will differentiate. The image that you're seeing here at the bottom is an image of a worm that has been studied pretty extensively because it's a pretty simple animal. It's easy to manipulate and grow in a lab. This is called C. elegans. And you can see kind of the tissue structure of this is also pretty simple, but shares some things in common with what we see in our bodies. We have a digestive system, we have reproductive structures and organs and cells, we have muscle tissue, we have an outer kind of covering similar to a skin, which they call a cuticle. But what we see here is different types of systems that start to develop based on stages in the embryo. So we've got over here nervous system is going to develop by the 5th cell division. We start to differentiate and create that nervous system. The outer part of the embryo eventually becomes by the 8th cell division, part of this cuticle or the outer covering of the organism. And then here we have 9 or 10 cell divisions. We get the feeding organism or organs that start to differentiate here, and that's the very internal part of this structure. Okay, so how this all happens is based on gene expression. And so when we talk about differentiation or cells obtaining roles, that's going to happen through the expression or the showing of different types of genes. So even though all cells in the body should contain the same DNA information, the same number of chromosomes, the same set of chromosomes from mom and dad, how we show that genetic information, how we express our genes is going to be different for different cell types. Okay, so for example, in the human body, a bone cell does not look like a muscle cell, and a muscle cell does not look like a skin cell, and a skin cell doesn't look like a neuron. And all of those structures and shapes and diversity of function, all of that is coming from which genes are being expressed. So in a bone cell, we only express this set of genes over here. And in a muscle cell, we express this over here, and so on and so forth. And so the stuff that we don't need to express, we don't get rid of. It's just we don't actively read that in the nucleus. Okay, it's not actively being kind of read off and used to create proteins. Okay, so differentiation again happens starting in the embryo based on cell location and based on combinations of genes that are being expressed or read and made into proteins. Okay. So looking at this whole process, like I said, this happens in embryonic development, but we also have this added component here of something called stem cells. So in order for a cell to differentiate and actually obtain a job or a role, it has to kind of start somewhere, right? And stem cells are going to be our starting point. This is kind of similar to you guys sitting in my classroom, right? You are kind of at your starting points. You're trying to figure out your pathway, where you're going to head and what your goals are going to be. The same applies with cells, right? Stem cells are kind of similar to that. So stem cells are a type of body cell. They're unique. They have the same number of chromosomes, same genetic information as every other body cell in your body. But they are kind of the initiators. They are where we start, okay? They are completely unspecialized, which means they don't have a job. They can remain unspecialized and undifferentiated, like you see right here, for a very long period of time. They can develop into lots of different things if pushed in that direction, okay? And they can also divide and renew themselves kind of forever. So stem cells have the ability, this really kind of unique ability, to become whatever it is we need them to become. And so for that reason, in our bodies, they become very, very important, because if we need more heart muscle cells, well, we have a heart muscle stem cell reservoir, and those stem cells in the heart muscle can be coaxed into becoming more heart muscle cells. All right, and so we see that process happening right here. If we go this route towards the bottom of this diagram, one stem cell, when it divides, will create one new stem cell, so it kind of replaces itself, and then also a specialized cell that becomes whatever is needed. Okay, we also have the ability with stem cells to just divide them in half and create 2 new stem cells through mitosis. So we can take these different pathways based on what's needed in a multicellular organism. And again, I'm talking about humans a lot, but again, recognize this is something that we see kind of across the board in many different species. All right, so we have 3 different types of stem cells, and we're going to talk about them in order from, I guess I should say, least specialized, earliest to latest or most kind of specialized, if we talk about stem cell options at least. So totipotent is the first word there. That's how you say it. Totipotent stem cells can become anything that's needed in the whole body. Okay, a totipotent stem cell is only what's present in a fertilized egg. That's it. So when we talk about fertilized egg, we're talking about sperm and egg having met. And when that occurs, you end up with a single totipotent stem cell. And if you think about this, every single one of us was created by that first fertilized egg. We all started as one single totipotent stem cell. However, once we go from totipotent and start to divide that single cell and start to create more diverse cells that are eventually going to make up a human body or any other organism, we lose the ability to still be totipotent. So we can create more stem cells, but the issue lies in it can't become just everything, right? They're going to kind of narrow their focus in terms of what they can actually become. So the only time we have totipotent stem cells is when we were a fertilized egg, okay? Pluripotent stem cells are a little bit more narrow in terms of what they can become, but they can still do lots of different things. Pluripotent stem cells are what we find in embryos, all right? So this is an example of an embryo here and embryonic stem cells right here in culture. And what we see is, again, I mean, if we're talking about the embryonic stage, look at this is not very human-like, right? I mean, we're talking about literally like a bunch of cells kind of stuck together without a lot of direction in terms of what they need to go off and do. So these are our pluripotent stem cells, and they can become, again, almost everything. The only thing, the only caveat, the only exception to this is that they cannot go backwards in time and go back and become a totipotent stem cell. All right, they have to only remain pluripotent, create more of their kind, or divide and differentiate and become something specific. But they can't go backwards and become totipotent, just like we can't revert back and become a fertilized egg. We can't revert back and become an embryo, right? So as we think about this, all these different types of stem cells are forming through development, all right? And you don't have the ability to reverse that process. So totipotent stem cells, again, one more time, are fertilized eggs, and that's it. Pluripotent stem cells are what we have in embryos during early organism development. And then we have one additional type of stem cell here, which is called multipotent. So multipotent stem cells are what you and I have in our bodies right now. We call them also adult stem cells, even though to have them doesn't mean that you have an adult. Like my 3-year-old has multipotent stem cells. It simply means that these are what we have available to us after we're born. All right, so these have a limited potential. They can become lots of different things. However, they can really only become cells of a related tissue. So unlike embryonic stem cells, which could form every tissue of the body, multipotent stem cells can only become tissues of a related type. All right, so if we have stem cells from the bone marrow here, okay, they can become red blood cells, they can become white blood cells, they can become platelets and lots of other things, but they do not have the ability to go off and become part of the lung or become part of the brain. So they're a little bit more limited, more narrow in focus in terms of what they can actually do. We also only have them in specific areas of the body. So we've got them in bone marrow, we have them in our brain, we have them in our heart, in our skeletal muscle, and so on, but they're not found everywhere. So our options, again, are kind of limited in terms of, how we can actually use these and create what's needed in the body, okay? So what that leads us into here then is this last little part about how this becomes kind of an issue when we think about the political realm, right? Social issues outside of, a body specifically. So stem cells, because they have the ability to do so many different things, have been an area that's been actively used in researching things like cancers, researching things like just general development and genetic disorders and lots of different things that we still have so many questions about. What we tend to use, what researchers tend to want to use, are either totipotent stem cells or pluripotent stem cells. So either those from a fertilized egg or those from an embryo. But the issue that we tend to run into is that embryonic stem cells and fertilized eggs are almost impossible to get. Like truly made, genuine fertilized eggs or embryos are not accessible most of the time. So what we typically resort to are donations from people that have gone through fertility treatments where embryos have been created basically in a lab, but were not used by the couple to create offspring. And that's kind of where we stand with that. And so the issue with that is that there are some folks that don't view that as being an appropriate use of an embryo, right? Embryos, if we view them as being humans and human beings, and that they have the potential to become a human being. Essentially, if we're using them for this type of research, we're not allowing them to do that, because once you remove embryonic stem cells from an embryo, the embryo gets discarded. It can't continue to grow. And so even though there's a lot of potential here in terms of research and scientific understanding, stem cells are kind of an iffy area in terms of research. We don't generally get a lot of political support and government backing from the federal government, because this is something that is kind of risky in a way. It's not something that everyone would approve of, okay? But here are some of those benefits for using those pluripotent stem cells. We can treat leukemias and lymphomas. We can develop pharmaceutical drugs and research processes. We can find cures to diseases and replace damaged organs using organs created in the lab from a patient's own stem cells, which are super cool. And so one of the things that goes along with that is this potential to also use adult stem cells. So you've got the pluripotent side of things, where we're removing those from an embryo, from a viable embryo, to be used for research. But then you've also got those reservoirs that I said we all have in our bodies, these adult stem cells, these multipotent stem cells that can be used theoretically to create organs for that one individual. So let's say, for example, I have issues with my pancreas, right? And within my pancreas, my cells are just not producing the insulin that's needed to help regulate my blood sugar. Well, the idea would be, if I have adult stem cells that can be coaxed into becoming pancreatic cells to create a pancreas, a whole new organ, then you could create that in the lab and then donate that back to the individual from which those stem cells came. So essentially, you run the risk of organ donation and rejection just completely out of the window. It's not going to be something that's even a possibility when you're talking about cells that are coming from that individual patient. And so that's a really cool area where we could be using these stem cells. In addition to that, we've also been able to scientists have figured out how to take multipotent stem cells and kind of turn them back. They turn back time, which I remember I said we can't do that naturally, but they've figured out a way to turn those cells back around and coax them into becoming what look like pluripotent stem cells, which are actually going to function like pluripotent stem cells. So what we've done is we can take specific genes, and they've introduced them into skin cells, just regular old skin cells. And those genes end up kind of turning those cells back into embryonic stem cells, back into those pluripotent stem cells that we so want to try and do scientific research. And if you can do that, then you don't even have to deal with that controversial kind of issue that comes along with potentially destroying viable embryos, right? Because you're essentially creating embryonic stem cells from regular skin cells from individuals that maybe want to donate to this research, okay? So there is some controversy associated with this. We could talk about stem cell research for a very long period of time, but in terms of what you need to know for your test, that's about it. So these here on this slide are called induced pluripotent stem cells. That's maybe a fourth category of these, if you want to consider it that. But again, essentially, they are regular cells that have been turned into pluripotent stem cells. All right. So again, your four types: totipotent, which is a fertilized egg; pluripotent, which is an embryo and embryonic stem cells; multipotent, which are adult stem cells present in you and me; and then induced pluripotent, which are cells that have been modified to function like pluripotent embryonic stem cells. Okay. All right. That's all I have for section 11.4. So if you have any questions or need anything more, again, feel free to reach out. Otherwise, that's all I got. Bye, guys. See you next time.